It is now my distinct pleasure to introduce two people who are supporting our speaker today, Marina Court and Yue Yu. Marina Court is a product manager for immunoassays at BioLegend and Revity, where she oversees the development and promotion of innovative tools that support cutting-edge immunology research. She earned a PhD in biomedical sciences from Cedars-Sinai Medical Center, where her research focused on cancer immunology. She also has hands-on experience in antibody and protein development and has contributed to biotech consulting initiatives through Biotech Connections at LA in the US. Yue Yu is a PhD student in Katrin Suter's lab at the University of Duisburg-Essen in Germany, with a research interest in infectious diseases, working on dissecting interferon alpha subtype-specific antiviral mechanisms against SARS-CoV-2. And with that, Marina, the floor is all yours.
Thank you so much for that great introduction. Hello, everyone. Good morning, good afternoon, and good evening. My name is Marina Court, and I'm the product manager for BioLegend and Revity for our immunoassays. BioLegend creates world-class cutting-edge antibodies, immunoassay kits, and reagents across a diverse set of applications for immunology and cancer, with over 30,000 trusted reagents and over 100,000 citations and growing. BioLegend is making an impact in labs across the world, and head over to biolegend.com to learn more about our products. Today, I have the distinct pleasure to introduce our speaker for today's webinar, Dr. Katrin Sutter. She is an associate professor at the Institute of Virology, University Hospital Essen, University of Duisburg-Essen, Germany. She studied biochemistry at the University of Frankfurt and earned her PhD in biology in 2010, followed by habilitation in virology in 2019. And since 2018, she has led the Sino-German Virtual Institute of Viral Immunology. Her research focuses on type 1 interferons, immunotherapy, and host immune responses to viral infections, including HIV, HBV, and SARS-CoV-2. Dr. Sutter is a recipient of multiple scientific awards, including the Loeffler-Frosch Prize for the Society of Virology in 2025, and she has extensive teaching experience in virology, immunology, and infectious diseases. And with that, I will turn it over to Dr. Sutter to present her webinar today. Thank you.
Thank you very much, Marina. Okay, can you see my slides? Yeah. Okay, perfect. So, dear Marina, the organizers from Bioprotocol, BioLegend, the audience, I would like to welcome you all to my presentation dealing with type 1 interference and immune responses. So my name is Katrin Sutter, as Marina already mentioned, and I'm a group leader at the Institute for Virology in Essen in Germany. So maybe Essen is not every one of you is familiar with Essen. So Essen is located in the western part of Germany that you can see here, and it was in the past very popular for coal mining. So, but let's talk about type 1 interference. So during viral infections, these viruses can trigger innate immune responses, including the production of type 1 interference, which you can shortly see here. And these then drive the expression of interferon-stimulated genes with diverse biological activities. So once the type 1 interferons are induced, they can bind either autogrin or paragrin to its receptor, which is composed of two subunits, IFNA1 and IFNA2, and this then activates the JAK-STAT signaling pathway, which you can see here. And then upon activation of the signaling cascade, we have done the expression of hundreds of so-called interferon-stimulated genes, or short ISGs, with various biological functions like antiviral, immunomodulatory, or antiproliferative. So in the human system, the type 1 interferon family consists of interferon alpha, beta, epsilon, kappa, and omega. They are located on chromosome 9. However, the system is more complex than it first appears. So interferon alpha is not a single entity. There are multiple interferon alpha subtypes. And this is a phenomenon which you can which is observed across all vertebrates. So the human system, for example, encodes for 13 different genes and 12 different proteins, whereas mice encode for 14 different interferon alpha proteins, and all these genes for mice are located on chromosome 4. So importantly, and this is really important for all of you to know, is that the human and the murine interferon alpha subtypes, so the nomenclature differs. So meaning that the mouse interferon alpha 2 is not an homolog of human interferon alpha 2, and this is also seen here by sequence identity of their protein sequences. So this is really important. So the numbers are different, and alpha 2 in human is not the same as alpha 2 in mice. So as I already mentioned, all type 1 interferons, they bind to the ubiquitously expressed receptor IFNA1 and 2, activates the classical JAK-STAT signaling pathway, and then hundreds of ISGs are induced that orchestrate the host immune response. However, the interference signaling is not that simple that it first appears. So in the human system, it is already known that all type 1 interferons have different binding affinities to the both receptor subunits, and that strongly influences their biological response. Furthermore, they can also activate non-canonical pathways, shown here, that regulate and also fine-tune their biological effects. So we and others have shown here that the different murine and human interferon alpha subtypes exhibit different antiviral properties. For example, HIV is strongly, so here you can see HIV is strongly inhibited by interferon alpha 14 or interferon alpha 6, whereas in other infections like SARS-CoV-2, human interferon alpha 5, and this is shown here in dark blue, strongly has a very high antiviral activity against SARS-CoV-2, whereas it has almost no antiviral activity against other viruses like HIV, HBV, HCV, and so on. So apart from their direct antiviral activity, interferons can also strongly modulate immune responses like NK cell, activation, maturation of dendritic cells, and also T cell responses. So in the past, we have intensively analyzed NK cell responses, cytotoxic CD8 T cell responses upon stimulation with different interferon alpha subtypes. But today we will focus on CD4 positive T cells and how they are shaped by different interferon alpha subtypes. So the field, so the interferon field, long assumed that all interferon alpha subtypes are functionally identical. So this is just a matter of dose, that because they all bind the same receptor, meaning just give more interferon of the one interferon or of the other, and then you see the same effect. So, but this phenomenon of different ligands binding to the same receptor having different outcomes is well accepted for chemokines. I mean, most of you know that there you have one receptor, different chemokines can bind to it and have different biological functions. And today I hope I can show you that also the interferon alpha subtypes, although they share some common functions, that they also exhibit unique properties. So as nowadays it is unfortunately impossible to order all recombinant murine interferon alpha subtypes. So you can buy all human interferon alpha subtypes, but if you go into other species, that's impossible. So therefore we produce them all in-house. So we overexpress, so we have plasmids expressing the different interferon alpha subtypes. We overexpress them in Hector-9-3 T cells. Then we collect the supernatant for up to 10 days. We concentrate the supernatant, so interferons have a size of 19 kilodalton, so we use 10 kilodalton filters that everything smaller than 10 kilodalton just flow through the filter. Then finally we aliquot and freeze those interferons. And to measure the activity of the produced interference, we use a reporter cell line, which is called an NIH3T3 ISRE luciferase reporter cell line. And these reporter cells harbor the luciferase gene under the control of the interferon-stimulated response element. This is here. where the complex, so the signaling complex, binds to. So this means when upon interferon binding here on the surface to the receptor, the classical JAK-STAT signaling is activated, and then this then leads to the transcription of the luciferase gene. And once the luciferase is expressed, you can then lyse the cells, because our luciferase stays in the cell, it's not secreted. You lyse the cells, and then you can just add a substrate, which is luciferin, and this is then catalyzed by the luciferase to oxaluciferin, and during this process, energy is emitted as light. And then you can just measure this using a microplate reader. So we have here Tecan Spark, and then we measure this and normalize it to recombinant interferon that we just buy from company with known activity. And then we have like a standard curve, and then we know the activation of our interference. We do also the same with with human or with swine interference that we are also producing. And with our in-house produced interference, we then wanted to establish an in vitro CD4 positive T-cell proliferation assay. And for that, we isolated the bone marrow of wild-type mice. In our case, those so naive wild-type mice. In our case, these were B6 mice. And we differentiated the bone marrow in the presence of IL-4 and GM-CSF for 7 days in cell culture. And then after these 7 days, we had mature bone marrow-derived dendritic cells. And these cells were then loaded for 2 hours with a viral antigen. So here we had an envelope peptide from a mouse retrovirus. It's called friend retrovirus, and I will come to that later in my presentation. So I will explain the virus a little bit more at the end of my presentation. So now we have the dendritic cells. We loaded them with antigen, and now we also need the corresponding T cells. So for that, we have a transgenic mouse model where with CD4 positive T cells that are transgenic or the T cell receptor is transgenic for this viral peptide. So we isolated them using Mojo sort from BioLegend, and this is quick and easy technique where you can enrich naive CD4 T cells by a negative selection. So it means you add an antibody cocktail with biotinylated antibodies that are targeted against all other immune cell subsets like B cells, granulocytes, CD8 positive T cells, NK cells, and so on and so on. And then you add nanobeads, which are magnetic, and then you just put your cell suspension into a magnet, and all cells that are then bound by an antibody, they are then moving towards the magnet, and then you can just separate the untouched target cells from the touched cells that you don't want. And then this is what you can easily see. I mean, this is now a picture from BioLegend website, but you can easily enrich the naive CD4 T cells using the Mojo sort magnet. And this is just like around half an hour, 45 minutes. It's really very quick. So now we have the T cells, we have the dendritic cells, but as we are interested in a proliferation assay, we want to track the division of our isolated transgenic CD4 T cells. So therefore we used a Tecan Violet. This is a dye which is similar to CFSE, which maybe some of you know, and this dye can passively diffuse into the cells. And then there's like the esters on this dye are then cleaved within the cell, and this leads then that the dye can covalently attach to intracellular proteins. And then you can just track those cells for quite a long time. And then when these cells divide, and this is what you can see here, they also pass the dye on to their daughter cells in lower concentration, and this phenomenon can then be easily visualized by flow cytometry. So we have our bone marrow dendritic cells, which we loaded with infant virus envelope peptide. we have our isolated, naive CCR transgenic CD4 T cells. And then we added our different mouse interferon alpha subtypes that we produced in-house and put that all together for up to 72 hours. And then we were interested in the proliferation of those cells, in the effector functions, and how the different interferons modulate the T cell response. So, and I mean, I've just explained to you how this TACIT violet is working, and this is then how it looks like. So you have these different peaks, and every peak represents one division step. So meaning that on the very, very right here, where you have the highest TACIT violet concentration, these are the undivided cells, and then every other peak then on the left side means 1 division, 2 divisions, 3 divisions, and so on and so on. So then we analyze how often do those cells divide, or do these T cells divide, and this is what you can see. So when we do not add interferon, which is shown here in green, we see that almost every CD4 T cell in our co-culture divides, whereas when we added like interferon alpha 2, interferon alpha 5, or interferon alpha 11, we have a significant reduction in the proliferation of those T cells, meaning that these interferons have a strong antiproliferative activity. For all the others, we saw either a minor effect, or also for some, like interferon alpha 1 or interferon alpha 6, they had no effect at all on the proliferation of the CD4 T cells. So if we have used viral antigen, and during viral infections, you usually induce Th1 cytokines, we had a closer look at Th1 responses. So therefore, we performed intracellular cytokine staining and analyzed the intracellular expression of, in our case here, interferon gamma, IL-2, and TNF-alpha. So what you can see is when we don't have any interferon at all, shown in blue here all the time, the overall intracellular cytokine expression is rather low. What's quite interesting for us is when we stimulate this co-culture here with interferon alpha 4, 6, 9, and 11, we do see a strong increase in cytokine and interferon gamma, IL-2, and TNF-alpha expression in those CD4 T cells. But what was quite surprising for us is that with interferon alpha 1 and 2 and 5, we did not see this effect. So there was no induction of interferon gamma, no expression of IL-2, and also TNF-alpha was rather low. So, and as naive CD4 T cells can also differentiate into other T helper subsets like Th2 cells, Th17 cells, regulatory T cells, and so on. We had a closer look if maybe other cytokines are induced upon stimulation with interferon alpha 1, 2, and 5. And for that, we performed a legend-plex. So what is a legend-plex? So this is a technique where you can analyze multiple effector cytokines in one sample. So up to 14 different cytokines or analytes can be assessed with very high sensitivity. And the very good thing is, especially when you're working with mice, you only need 25 microliter of your sample. So especially if you want to look in serum samples, or if you have 96 well plates and you don't have so much supernatant, then this is really cool because you only need very little sample. So the principle of this assay is that you have two different kinds of beads. So these are here results from me. So you have two different sizes in beads, A and B, and depending on their size, you can just differentiate them in the forward-sideward scatter on the flow cytometer. And then these different beads are filled with different amount of APC concentrate fluorescence, and this is then what you can see here. And then all the steps are similar to an ELISA. So you add, then you analyte if your sample, and if you have, let's say, interferon gamma in your sample, it binds to those beads which couple the antibody against interferon gamma on the surface. And then you add again antibodies, then again against the different analytes. You add those are biotinylated antibodies, then you add streptavidin PE, and then you can easily measure this with any common flow cytometer. So you don't need any special devices, just it's very easy, just also short time. It's, I would say like 45 hours altogether, the assay. And we have done that pretty often in our lab, so we really like that assay. So, and these are the results for that. So we used here mouse inflammation panel because that perfectly fits for our results. And what was quite interesting for us is when you look at these two lines, that we saw a strong increase in IL-17A expression and also an IL-6 expression in comparison to the untreated co-culture after treatment with alpha-2 and alpha-1, and also here with alpha-5. So we had a closer look at this. So I have before never worked with IL-17 or Th17 cytokines because I'm from Virology Institute. We are not really interested in Th17 cells. So we are always looking only for Th1 cells. So then we started to do a lot of ELISAs. So we tested, maybe I could just go one slide back. We tested all the cytokines that are important for the differentiation of Th17 cells, and we also tested those cytokines that are produced by Th17 cells. So these are all here, so IL-17A, IL-21, IL-22, IL-6, IL-23, and TGF-beta. And what you can easily see that again here for IL-17A, we do see a significant increase after stimulation with interferon alpha-2. And what was also quite interesting for us, that we also saw a significant decrease after stimulation with alpha-11. So we saw the same trends here with IL-21. Also with IL-22, we saw that alpha-2, alpha-5, they had similar levels compared to the untreated ones, whereas like especially with alpha-11, we saw a decrease here in IL-22 levels. And also quite interesting, we saw increased TGF-beta levels after stimulation with interferon alpha-2. So then we also analyzed the intranuclear expression of transcription factors. So if I go maybe one slide back, I didn't mention that. So also every T helper subset is characterized by the expression of specific transcription factors, like in Th1 cells, we have Tbet, in Th2 cells, GATA3, and in Th17 cells, we have ROgamma-T. So you can easily do that by doing an antibody staining within the nucleus. And we stained these co-cultures for Tbet and ROgamma-T. And all the results I showed you up to now were at 72 hours post co-culturing. And we looked there and there was nothing to see. So we couldn't see any effect on ROgamma-T and Tbet expression. Then I talked to some colleagues and they told me, oh, the 3 days is really early for transcription factors. Just keep the culture going and look a bit later. And that's what we did. So we then checked day 5 of co-culturing, and there we saw significant effects. So we saw significantly more ROgamma-T positive CD4 T cells after stimulation with alpha-2 in comparison to alpha-11. And we saw the opposite for Tbet. So there, after stimulation with alpha-11, we saw significantly more Tbet positive CD4 T cells in comparison to interferon alpha-2. So what we saw is we saw a lot of IL-6 in our cultures. And so we thought, okay, maybe IL-6 is really important. So to further scrutinize that, we performed again our co-culture system. And now we added an antibody that blocks IL-6. And what was quite interesting for us is really, this is again what you see here in red is interferon alpha-2. And blue, dark blue is without interferon, and in light blue we have interferon alpha-11, and without any blocking antibody. So you see a lot of IL-17 expressing CD4 T cells after stimulation with alpha-2. If you add in the blocking IL-6 antibody, the IL-17A expression is completely gone. So it really seems like IL-6 is, in our case, a master regulator of the interferon alpha-2 induced IL-17 expression. So then we also said, okay, let's have a look on the polarization of these Th17 cells. So now we polarized the cells in vitro, so without our dendritic cells. So we isolated naive CD4 T cells, and there we really also got rid of all the regulatory T cells, so they are all CD25 negative. And then we had them in culture for 5 days altogether in the presence of the stimulation mix. And I just wrote it here because there are really a lot of different things inside. And then after these 5 days, we analyzed the expression of IL-17A, but also of TNF-alpha on those cells. So, and what was quite interesting is that we saw a significant reduction again with interferon alpha-11, so that nicely fits to the data that we've seen before. And we also saw a significant increase in TNF-alpha expressing CD4 T cells after stimulation with interferon alpha-11. However, for interferon alpha-2 here, that was not significant, and you see the effect is rather low here. However, we also tested the supernatants of the T-cell polarization assay here. And what you can see, so here without interferon, alpha-2, alpha-5, alpha-11, if we had a look at IL-6 and IL-17A, which is here, you see a strong increase here after stimulation with alpha-2 in comparison to the unstimulated controls. Okay, so I just want to sum up this very first part. So I could show you that all the murine interferon alpha subtypes here exhibited distinct properties on CD4 T cells. We observed that interferon alpha-2 and alpha-11, they both could strongly inhibit the T cell proliferation, whereas other subtypes had minor effect or even no effects on T cell proliferation, like interferon alpha-1 and interferon alpha-6. Interestingly, interferon alpha-11 strongly promoted Th1 responses, but suppressed Th17 responses, which I could show you by cytokine expression in ELISA, Legend-Plex, and also intracellular FACS stainings, as well as intranuclear detection of the transcription factors TBET and ROGAMA-T. And in contrast, the stimulation with alpha-2 and also partly with alpha-5 did not induce a Th1 response, whereas it promoted more Th17 response. And this was strongly induced by the induction of IL-6. And when we block IL-6, we could completely prevent the induction of the intracellular IL-17 expression. So to potentially gain a mechanistic insight on the biological differences between the interferon alpha subtypes on our specific CD or virus-specific CD4 T cells, we next performed transcriptomic analysis. So for that, we have again our assay. We kept the cells for 72 hours again, all the same as before, and then we did sex sorting, where you can then, you just use antibodies and then you can just sort your cells. Then we have the dendritic cells in one vial afterwards and all the T cells in another vial. Then we isolated the RNA of the dendritic cells and also CD4 T cells, and then we performed RNA-seq using Illumina NextSeq 500 platform. So today I will only focus on the CD4 T cells, but of course we have also analyzed the bone marrow-derived dendritic cells. So we observed clear differences in the numbers of up and down regulated differently expressed genes among the different groups. So what you see here is the simulation with interferon alpha-11 resulted in more than 700 differently expressed genes. Almost half is up, half is down regulated, whereas stimulation with alpha-2 and 5 resulted in only around 140 and around 90 differently expressed genes. And what is also nice here is that we saw interferon alpha 11 also down regulated IL-17A expression, which nicely confirmed our previous data. Yeah. So, and when we have a look at the different genes that were up and down regulated, there were genes like here we have serpin B9B, we have AVMET, we have GREC-CD, I5, which is here, I5-213 and I5-214. These are genes that are associated with negative regulation of apoptotic processes, metabolism, restriction of proliferation, also antiviral responses, so especially these here are antiviral genes. So we then also analyzed the distinct and common genes that are regulated by these three different interferon alpha subtypes. So as expected, we identified here only, so we have 21 common differentially expressed genes, and they were all associated with defense response to viruses and innate immune responses or regulation of the helper cell differentiation. So including genes like so we have here ISG15, ISG20 down here. We have IFI27, IFNM2 all here. So this was something, okay, classical antiviral genes. This is what we expected. But what was quite interesting for us is that the majority of the genes, they were specific for either alpha 11, for alpha 2, or for alpha 5. Yeah, so there were not so many common genes. And there is an interferon database where people uploaded all their data when they performed stimulations with different interferons in different tissues and so on, and then all the different interferon-stimulated genes. They are all catalogized there. And then we compared our differentially expressed genes with the database, and we saw that at least for interferon alpha 11, around 2/3 of the genes were mentioned in the interferon database, whereas for 2 and 5, especially for alpha 5, that's almost half of the genes were not mentioned in the interferon database. So then we used all those genes here in dark blue, which are also within the interferon database. And then we performed a gene ontology pathway analysis of these differentially expressed genes to figure out so which pathways are differentially modulated by these interferons. So, and what we saw is that there were, especially for interferon alpha 11, many pathways like cellular processes, which are shown here. Then we have biological regulation, we have metabolic processes, localization, and so on, that are solely activated here in the case of interferon alpha 11, but almost nothing here for 2 or 5. We also then performed string analysis with all the genes that were up and down regulated after interferon alpha 11 to figure out if we see any networks or clusters. And these clusters were connected to, again here, RNA processing, metabolic processes, mRNA processing, and so on. So therefore, we became interested in the immunometabolism, and we also have a group, and this was all done in cooperation with a very talented assistant professor at our university who was focusing on immunometabolism. And so we were interested, okay, what's going on in the CD4 T cells? Has the stimulation with interferon maybe an impact on immune metabolism? So, and if you think about T cell activation, these are, there's an aerobic glycolysis, this is induced, and I mean, during activation, the T cells just need more metabolic demands to get activated and so on. So, and these alterations in metabolic processes, which you can observe in CD4 T cells or also in others, there is most of the energy is either coming from glycolysis, which is shown here, or it's coming from oxidative phosphorylation, which is located in the mitochondria. And both pathways lead to the production of ATP, which is required for like activation and all this stuff. So we were interested from which pathway is the energy coming from. You might say, okay, but there are also other pathways. We know we only check those two. Until now, we haven't analyzed amino acids or fatty acids that might also be important, but we have mainly focused now on glycolysis and oxidative phosphorylation. You can do this by different assays, and we also tested different assays. So one is the seahorse assay, but you can also do SANET analysis where you use antibodies against puromycin, and then you can look on a single cell level, but here we use seahorse. This is an assay that utilizes differences in proton and also in oxygen within the culture. And you add different kinds of inhibitors to the culture, which block different steps of the different pathways. Like you can add 2DG, which then blocks the glucose metabolism here, or you can add like oligomycin, which inhibits the ATP synthase as part of the oxidative phosphorylation here in the mitochondria. And from that, you get a lot of information where you figure out which pathway is more important. Is it more glycolysis or is it more oxidative phosphorylation? So Elisabeth is more an expert on that. She helped us with all these analysis. But what was quite interesting for us is so we examined different things. So we used an assay which is specific for T cells in this seahorse technology. The first thing what we did is we examined the oxygen consumption rate, and this is like an indicator of mitochondrial respiration. And then afterwards, we also analyzed the extracellular acidification rate, and this is an indicator of glycolysis. So what you see here, so first we looked for the oxygen consumption rate, and then depending on the height here of the peaks, so how high they are and then how they go down, you can just calculate different things. And I don't want to go too much into detail, but what you can definitely see is that mitochondrial respiration is really important for stimulation with interferon alpha-11. So this was strongly increased, all these like basal oxidative phosphorylation, reserve capacity, maximum respiration, and so on. All these things were significantly upregulated after stimulation with interferon alpha-11 in comparison to stimulation with interferon alpha-2 or to either the unstimulated ones. So it doesn't look like the glycolysis is important. So what I want to tell you here is, so from our data, it looks like the energy that the T cells need in our cold culture are more coming from oxidative phosphorylation and not from glycolysis. And it definitely looks like the T cells behave completely different to alpha-11 and alpha-2 treatment. So it looks like, especially with alpha-11, they get a lot of ADP out of that, whereas with interferon alpha-2, they more look like in an energetic state. So to sum up the second part, so what we see is we see qualitative differences in ISG expression with interferon alpha-11 inducing much more differently expressed genes than alpha-2 or alpha-5. And furthermore, we also think that the oxidative phosphorylation is the major ATP producing metabolic pathway in interferon alpha-11 stimulated CD4 T cells, whereas stimulation with interferon alpha-2 led to a more energetic metabolic state in CD4 T cells. So, and then last, we went, so these, all the things that we have seen before were all in cell culture. I mean, that's nice. We see effects, we have different interference. One is producing more Th1 response, one is producing more Th17 response, but I mean, this is really concentrated in a small well. You only have this kind of cells there, no diverse cell subsets, and you really have one single interferon there. So therefore say we have to try this in a complex in vivo situation. So we went into the mouse, and there we have the friend retrovirus complex. So this is a mouse retrovirus that our institute is using for many years. And this is an oncogenic gamma retrovirus complex of mice. So it consists of two different viruses. So we have the friend murine leukemia virus, which is apathogenic, and it's an helper virus. And we have the spleen focus forming virus, which is pathogenic. So I don't want to go too much into detail. But what we do with this virus is, for us, this is a very nice model to analyze retroviral infection. And if you think about retroviruses, you should definitely know like HIV. And you can see also here the target cells, and then you would say, okay, this is completely different to HIV. Yes, that's right. And also the pathogenicity is different to HIV. But the immune response during friend virus infection is really similar to what people have observed in HIV infection. And what is also quite important is that it really depends on which kind of mice you're working. So if you use B6 mice, and that's what I'm doing most of the time, these mice are quite resistant against the infection, meaning they are not really susceptible to friend virus induced disease. So they do not develop splenomegaly, and they do not develop lethal erythroleukemia, because these mice really have a very strong immune response, but unfortunately they cannot completely eliminate the virus, so the virus still persists. So we used those mice, and then we infected B6 mice with friend virus. And then at day 5 post-infection, and this is the time point where APCs, so antigen-presenting cells, start to interact with the T cells. So then T cells start to proliferate, get activated, and so on. There we started to administer interferon, either alpha-2, alpha-5, or alpha-11. So, and then daily, because the interferons have a quite short half-life time. So our interferons are not pegylated at the ones that you can find in the clinics. So therefore we have to inject it every day into our mice. And then of course we had also untreated mice as control. So, and then after 5 injections on day 10, we sacrificed the mice and analyzed viral loads in spleen, analyzed Th1 and Th17 responses, and also looked for cytokines in serum. And what was really nice for us, that when we treat the mice with interferon alpha-11 here, you can nicely see a significant reduction in viral load, which was not the case when we treat the mice with alpha-2 and alpha-5. And then what was also quite nice is here you can see that when we treat the mice with alpha-11, we saw a significant increase in interferon gamma, so Th1 responses, whereas with alpha-2, we saw again a significant increase in IL-17 producing CD4 T cells. And so we also analyzed the blood of these mice, and also there we saw that more IL-17A and more IL-6, so systemically, was induced after treatment with interferon alpha-2. So so what we have seen in our in vivo infection is that interferon alpha-11, also in a very complex system with all immune cells and so on, we could see that interferon alpha-11 enhanced the antiviral Th1 response, significantly decreased friend virus replication in vivo. So we do see here an antiviral activity, while the application of interferon alpha-2 completely failed to control the viral replication, and it induces a more pro-viral Th17 phenotype in friend virus infected mice. So finally, I hope I could convince you that not all type 1 interferons are the same, and especially the subtypes. So we didn't talk today about beta, but we also worked a lot on beta versus alpha. So all these subtypes, they have really qualitative differences in terms of their immune metabolism, of their ISG expression, of their antiviral activity, and their immunomodulation. And I hope that after the presentation, you will remember for a lifetime that there's not only one alpha, we had a lot of alphas. And especially if you're working on innate immune responses, type 1 interference, you should always keep in mind that if you measure an interferon by ELISA, this is only part of the response. So if you only measure alpha 2, this is really only part of the response, and maybe a lot is missing. So with this, I would finally thank the organizers for giving me the opportunity to present our interferon research here. I would just like to thank all my colleagues that, of course, contributed to the work, and of course, you for your attention. Thank you.
Thank you so much, Dr. Sutter. That was a great presentation. Really insightful work into the different interferon alpha subtypes and how they, skew different T helper subsets and the metabolic differences between those as well. As it's a great story. To our attendees, if you have any questions, we already have quite a few in the chat, but feel free to keep putting them in the chat, and we will start to work through some of these here. First question from Anam is in regards to your CD4 T cell proliferation assay. Did you add IL-2 to the culture each day along with interferon alpha, or were the cells cultured with interferon alpha alone, relying on endogenous IL-2 production to support the proliferation? And what method did you use?
Very good question. So it's really only the bone marrow derived dendritic cells where we loaded the peptide, then we add the T cells, and then the interferons. So there was no IL-2 in the culture. And before we had IL-4 and GM-CSF for differentiation of the dendritic cells, but completely no IL-2. We are also trying at the moment to do these experiments in the human system, so with monocyte-derived dendritic cells, and then also with CD4 T cells that we isolate from Buffy codes. And there, of course, we are trying a lot, and there we are also adding IL-2. But in this 3-day culture, we didn't really needed additional IL-2. So, I mean, you also saw that we do induce IL-2 there, and that was completely enough. Yeah.
Mm-hmm. Okay, great. And you had just touched on it a second ago. We had another question, whether you've repeated these experiments using human samples. It sounds like you have. Do you have any insights on whether the mouse data is reproduced?
So we are currently working on that. And the other thing that we also have a mouse with the human interferon receptor on top, so that we can compare the human interference on this assay that we have, because this is working pretty well. And then on the other side, we are trying to establish this in vitro assay with human samples. So I cannot really say so much at the moment, but we definitely also see their differences when you use the different interferon alpha subtypes. And you cannot say it's alpha 2 or it's this, so it's really different to the mouse system.
Mm-hmm. Yeah, that makes sense. Based on the homology you showed earlier on too, I'd imagine there'd be some differences. Okay, another question. Do you think that, I'll just say alpha 2 and alpha 11 have antiproliferative effects on CD4 T cells, and different alpha 11 and other interferons have different stat requirements giving different results?
So we definitely saw some genes that are also important in proliferation of cells that were either up or down regulated after stimulation with 2, 5, and 11. And what is also known from the literature that this antiproliferative effect of the interference depends on p53. Yeah. I mean, this is not something that we further dived into, but I mean, this is a known mechanism that interferon alpha can activate p53, and this then leads to this antiproliferative effect. Yeah, and then for the stats, of course, this is something that we are currently doing. So we are looking for STAT1 phosphorylation, STAT2 phosphorylation, but also for others like 3, and then also 4 and 5. And we have published that with the human interference, that especially there, it's another one is alpha-14, which is quite potent in many viral infections, that can really induce a lot of genes that others like a human interferon alpha-2 cannot induce, and that really depends on STAT3 and STAT6 signaling. So this is something that we are also now looking in the cell culture, if maybe we do see there a lot of STAT3 phosphorylation, because also STAT3 is important for IL-17 and also for IL-6. So this is something that we are currently analyzing.
Great, thank you. All right, we have another question from Basha. Did you observe any toxicity upon interferon alpha treatment in the mice?
Good point. So, so for our when we just inject 5 days, we do not see anything. With the mouse model that I just mentioned before, where we have the human receptor on every cell in the mouse, we are now checking more the side effects by interference, because this is something that is known from patients that they really suffer from severe side effects by interference, like flu-like symptoms, but also depression, anxiety, and all the stuff. But the thing is, in patients who really take those interferon therapies for almost a year, like 48 weeks, and with these short-term treatments, we do not really see anything. So we are looking right now for fever. So we're checking rectal temperature in the mice. We are looking for cytokine storms, systemic, but also in the brain. So we are isolating different parts of the brain, like amygdala, and really looking for expression of IL-6, TNF-alpha, and so on, to really see if there are a lot of pro-inflammatory cytokines upregulated. But so at least I can say, if you only inject it for a few times, we do not really see any side effects in the mice. They behave totally fine. And I mean, especially with alpha 11, the viral load goes down, so the mice feel much better. Yeah.
Okay, good to hear. A question regarding your bulk RNA sequencing of the interferon alpha stimulated CD4 T cells and dendritic cells. Have you repeated those experiments using human samples?
We have also performed a lot of RNA-seq with human samples. So we have done a lot during SARS-CoV-2, where we stimulated cells with the different interferons. So these were, of course, lung cells and also primary cells from the lung. And then we looked there for genes. We have done a lot in hepatitis B virus infection, where we stimulated hepatocytes, primary hepatocytes, so cells from the liver, with human interference. And especially there, what we saw is that some interference can induce like genes that are known from type 2 interference, so interferon gamma. So, and it really looks like that some interference can stimulate different signaling pathways and then induce genes that are not coming for type 1 interference, more for like 2 interference. And we have tested now all the 12 human interferon alpha subtypes for this phenomenon, and we saw that half of them can induce like a gamma-like response, whereas the other half not. So they just, or mainly signal via STAT1, STAT2, but they do not upregulate gamma-sensitive genes. Yeah.
Our next question from Tony kind of builds off of this, moving into human samples. And his question was, how much variation is there in interferon alpha subtypes in humans? For example, was there more COVID deaths in alpha 8 negative samples?
So I mean also for the humans, so the sequence homology is similar like in the mice. So you have, let's say, 75 to 95 sequence homology within one species. So the first thing is that every cell in the body can produce every subtype. So, and I mean, of course, there are people that do have some SNPs in some subtypes, but I would say normally everyone can just express every subtype. I think something what we know from SARS-CoV-2 and what is getting more and more popular right now are autoantibodies against interferon alpha. And there people have shown that especially against the interferon subtypes, most of the autoantibodies neutralize the interferon subtypes, which is not the case for interferon beta or other type 1 interferons. But what people have shown is, especially in COVID-19, is if people have high amounts of these autoantibodies against type 1 interferons, they really suffer from severe infections. And also if you have any genetic disorders in the interferon signaling pathway or so on, also those patients really suffer from severe diseases.
Mm-hmm.
Yeah. So I don't think that there are any special patients that do not express only one subtype. Yeah, but this is something that UA is right now doing. I mean, she's trying to clone cell lines where we really knock out all the single, so all in every cell line, one single subtype that we can definitely see, okay, what happens when a cell line can express only 11 out of 12 subtypes. Does that have an impact on SARS-CoV-2 infection? Mm. So this is something that that UA is working on. Yeah.
That'll be really interesting. Okay, we have two more questions here. And this is kind of a combination of two questions that we got regarding whether there were differences in the effects for different interferon alpha subtypes for different ages and genders of the mice.
Oh, that's good point. There, I would say the model is not the best because in virus infection, we do not really see sex-specific differences. So normally I would say yes, mice or then female mice have or should have higher interferon responses and so on. So they do express more TLR4. But in our case, as we order our B6 mice, we only work with female mice. They are all quite young, so like 8 weeks, 8 weeks of age. So we haven't really analyzed that. But as I mentioned, so in virus infection, so we had a colleague, she really analyzed it in detail if there are differences in immune responses in female and male mice, and at least for this infection model, there are no differences. So then we should maybe go into another system. But yeah, definitely aging is a thing, but I have never analyzed things like that. But it's really interesting. Yeah.
Yeah, that would open up a whole new set of questions, I'm sure. Okay, our last question is, do the responses and the productions of the different cytokines differ depending on which type of viral antigens you use to load the bone marrow dendritic cells?
Very good question. So for the CD4 T cells, we haven't tested that, but before we worked with a similar model with CD8 T cells, and there we also had an virus peptide, but we did the same with an ovalbumin peptide. So then with that specific T cell receptor and with an influenza specific T cell receptor and that peptide. And there we really saw the same effect. So We don't think that this is really depending on the viral peptide. This is really, let's say, a global effect of the interference on the T cells. Yeah.
Okay, great. All right, I think that is all the questions that we have here today. Thank you so much to the audience for your participation and your good discussion. Thank you, Dr. Sutter, again for the very insightful presentation, and to Bioprotocols for hosting us today. Thank you all so much.
Thanks. Yeah, that was really great. Thanks for both an interesting talk and great discussion. And thanks everyone for being here.